Our next speaker is Madeline Lancaster, a group leader at the MRC in Cambridge, England, and a pioneer on cerebral organoids. And she will talk today on examining human brain structure and function with cerebral organoids. Madeline, please. Your microphone is still muted.
Oh, there we go.
Okay, now you're on, Madeline, please.
I couldn't see the buttons at the same time as seeing my presentation. Okay,
yeah.
So here we go.
Now we can hear you.
All right, good. So it's really fantastic to be able to tell you about some of the work that we're doing in my lab using brain organoids. So the main focus of my lab is trying to understand what features are unique to the human brain, in particular what features arise during development that give us our unique cognitive capabilities. And so primarily we're interested in human brain size and the developmental events that lead to our very large brains. So this is just a few scale images of different brains from human, gorilla, and commonly used animal model mouse. And basically, brain size is not only just, in absolute terms, very large in humans, but also when you take body size into account. And that's what this so-called encephalization quotient is showing. So basically what this number means is that our brain is around 7 times larger than it should be for the body size that we have. And so, of course, brain size is determined by the cellular makeup of the tissue. And so, for example, neurons are really quite dramatically expanded in humans, somewhere between 80 and 100 billion neurons in the human brain. So we're interested in how this really huge expansion occurs. And we're then also interested in how these cells then connect up with each other in the correct way, because of course those 80 to 100 billion neurons need to be connected in a particular fashion in order to make a functional network. This is not just a randomly connected mishmash of cells. So we're also interested in how these cells find their proper targets. And so to look at that, we are interested in development, and in particular human development. And so before I jump into brain organoids, I will just give a background on some of the key steps of human brain development that we're interested in. So these are sagittal section images taken from mouse embryos, human embryonic brains at different time points. And what you see is that the brain starts out as what's called neuroepithelium. So it's an epithelium, and just like all epithelia, it has a fluid-filled cavity on the inside and epithelial cells that line it. And different parts of the neural tube will expand at different rates, with the most anterior portion expanding really quite dramatically, and the dorsal portion of that giving rise to the cerebral cortex, which you can see here is quite large in humans. So if we were to zoom in on the wall of the developing cerebral cortex, we'd see a cytoarchitecture that looks something like this, where, like I say, the tissue starts out as an epithelium, and these epithelia take on a pseudostratified organization and are initially dividing symmetrically to expand their numbers. So that this basically sets up the founder stem cell pool that will then give rise to all the neurons. And then at a certain time point, those cells switch their mode of division and start to produce more differentiated daughter cells, including, for example, intermediate progenitors, outer radial glia, and neurons. And neurons then use this long basal process that these now so-called radial glial stem cells. So these are now the neurogenic, still epithelial in many ways, but because they also maintain this basal process that connects to the outside of the brain, the neurons then migrate outward using this process as a guide and position themselves in the cortical plate. And the cortical plate will become the gray matter of the brain. And then these neurons, depending on where they are and their identity, will also project in very stereotypic fashion to generate this beautiful thing called the human brain connectome. And so we are interested in various events of this process and trying to understand at each key transition what are the human-specific events that give rise to our very large and complex brain. So obviously this is a human question, and so therefore we need a human model. And so the model that I developed when I was a postdoc is what we called cerebral organoids. And these are essentially trying to model various brain regions, actually, as closely as possible to in vivo development. And so to do that, we actually have to go even further back in development and look at the very early stages of embryogenesis. And so this neural tube that I showed you actually forms from the neuroectoderm, which will basically form during gastrulation and will fold up in the midline and form this, this neural tube. So we wanted to try to model these steps as closely as possible. And so without going into too much technical details, this is just kind of an overview of what we came up with, which is basically to take pluripotent stem cells that are essentially these inner cell mass type pluripotent cells. We then form these embryoid bodies, which basically is just aggregation of these cells. They spontaneously start differentiating and forming different germ layers, similar to gastrulation-like events, although not as organized in a spatial fashion. But interestingly, we do see that neuroectoderm tends to form around the outside, and then when embedded in matrigel, these form these neural tube-like buds, which can get really quite large when we subject them to agitation. So we've made a few, I think, pretty major improvements to this method. To be honest, we don't actually use the original method in my own lab anymore. So, for example, we've now started using a bit of bioengineering to improve the reliability of the protocol, and we can get these really quite large and beautiful tissues that have these sort of lobular structures coming off. And each of these lobes is filled with a fluid-filled cavity, very much like those fluid-filled ventricles of that developing cerebral cortex. And so if we section them, we can see that these, like I say, these lobes are filled with fluid, and they have a very dense layer of stem cells here in red around that ventricle, which is just like in vivo. And you can also see that each of these lobes gets to be really quite large. So we look at these essentially like several sort of telencephalic vesicles, basically the primordium that's going to give rise to the cortex, forming off of these organoids. And so we can look at each one of these lobes as sort of a unit. And so then within those lobes, within the wall of that developing cortical tissue, we can see a very nice layering, very similar to the mouse cortex, for example. So we've made a few other improvements lately. So for example, we've been looking now more at later events in neuronal maturation, and we've been subjecting these now to air-liquid interface cultures culture conditions in order to improve neuronal maturation and survival. And we get really nice long-range connectivity and bundling, very similar to the kinds of tracts you would see in a developing brain. And we can also then hook up these tissues to other explant cultures. So for example, here we've done a co-culture with some mouse tissue, mouse spinal cord that also has muscle, and we can actually get innervation. And when we stimulate the human organoid, we can get muscle contractions of the mouse tissue. We've also recently turned to trying to model the human cerebral spinal fluid system, so the choroid plexus of the brain, which generates the cerebral spinal fluid. And so we've developed this method to make these choroid plexus organoids, which look very similar to the kinds of choroid plexus you'd see in vivo. And they form these very large fluid-filled sacs, essentially, that are filled with a fluid that looks very similar to in vivo CSF, and form a very nice barrier, just like the blood-CSF barrier in vivo, that doesn't allow many things across. It's very selective, but it does allow, for example, L-DOPA across, just like in vivo. And now, very recently, we've also used these organoids to try to understand a little bit more about the SARS-CoV-2 neurotropism, the virus that's causing COVID-19. And we got interested in this because we noticed that the receptor ACE2 seemed to be highly expressed in our choroid plexus organoids in particular, but not so much in our neural tissues. And in fact, we found that it very successfully infects choroid plexus tissue and leads to a breakdown of the barrier. which you can see here stained for claudin 5, which is marking these tight junctions between the cells. And you can see those tight junctions essentially dissolving after infection with the virus, stained here for the viral spike protein. And functionally, we also see a breakdown of the barrier with fluid leaking out of these organoids. And so this is work that we've just recently published showing that these cells are susceptible and implying that potentially if this virus does make it into the blood system, it could then target choroid plexus cells, which would be very bad for the barrier that's protecting the blood and CSF. So I really wanted to tell you, though, actually about a story that's completely unpublished. And this is work that's led by Eva Kilava, who's a senior postdoc in my lab, using organoids to try to understand something about human sex-related brain differences. And we got interested in this because it's been known for a while that there is a sex bias in a number of neurological conditions. So for example, ADHD and autism spectrum disorder show a male bias, whereas, for example, eating disorders and anxiety and depression show a female bias. And this suggests that maybe there could be some features about male and female brains that might already set them up different for a different susceptibility. And although on an individual level, there are not, you can't really tell male and female apart, on a population level, there is an average, males do have an average larger brain size compared with females, suggesting that there really may be something, setting up the male brain a little bit differently than the female brain. And indeed, there's been a bit of examination of gene expression in male and female brains over time, showing that actually the stage at which you see the most differential gene expression is actually during fetal development rather than postnatal or adulthood. So Eva and I got interested in this question and trying to maybe use brain organoids to understand what could be setting up some of these sex-based brain differences. And there are essentially two possibilities. So one is that it's sex hormones that are playing a role in the brain. And two is that it's actually the sex chromosomal makeup that's intrinsic to each cell in the brain that is setting up these differences. And there's data from fetal development that suggests that hormones could play an important role in this process, because during development, soon after actually the testes in male embryos has formed, there's a very large surge of testosterone in the male embryo that's almost as high actually as the testosterone surge during puberty. So it's really quite a large amount of testosterone being produced by the male testes during development. So that would suggest that there may be high amounts of male hormones in a developing male embryo. So basically we wanted to look at this question. So we took stem cells from, we took two cell lines, basically, a male and a female line, and then we exposed them to different sex hormones, male and female sex hormones. So testosterone and dihydrotestosterone, or DHT, are androgens, male hormones, and estrogen is a female hormone. And then we just looked at various different features of the organoids to try to understand what types of differences were present. And we chose a time window at which the organoids match morphologically the time window when this surge would be happening in vivo. So basically, we did a whole panel of analyses, and I'm just going to jump to the differences we saw. And basically, the most striking difference we saw was actually on these intermediate progenitors, which are these green cells here I've labeled in this diagram, which are a trans-amplifying cell type that will then give rise to more neurons. And what Eva found was that when she treated with androgens, she saw a very large increase, not a very large, she saw an increase in these TBR2 cells, which is not a huge increase, but it is significant, and this is quantified here as well. And this was regardless of the cell line that we used. So whether we used male or female cell lines, we saw the same effect. And importantly, we didn't see a difference between male and female cell lines under control conditions. So suggesting that the chromosomal makeup is probably not making a really big difference here, but that sex hormones might be having a pretty strong impact. We then wanted to understand where this increase in intermediate progenitors was coming from, and so Eva did some lineage tracing here by using a virus to label individual clones of radioglial stem cells, which give rise to those intermediate progenitors. And she was able to show that clones after treatment with DHT were larger. And also, if she used an alternative approach, which is to use a constitutively active androgen receptor, she saw again that the clones were larger and that they were cycling more. So this shows that androgens seem to be promoting the proliferation of the initial radioglial stem cells, which will give rise to those intermediate progenitors. We then did RNA-seq to try to identify what genes were responsible for these differences. And a number of pathways came out of this analysis, but in particular, chromatin remodeling factors and HDACs came out. And so we looked in particular at HDACs by using an inhibitor of HDAC activity called valproic acid. And actually what we found is that when we treat with valproic acid, we see the opposite phenotype to what we see when we treat with androgens. So we see a decrease in these TBR2 cells. And this can be rescued with androgen treatment, showing that these are in the same pathway indeed. And so this suggests that HDAC activity, like the androgens, is promoting this stem cell proliferation, and that it may be downstream of the testosterone. And this is really interesting given that, of course, a number of these sort of sex-biased conditions are also thought to involve some of these sort of epigenetic chromatin remodeling events. And actually, there's an interesting paper showing that the social deficits in the SHANK3 mouse model of autism could actually be rescued by HDAC inhibitor, which is VPA, which is what VPA is doing. So this could give us some insight into the sex bias of autism spectrum disorder. We also noticed human phenotype ontology terms associated with brain size. And as I mentioned at the beginning, of course, there's also on a population level differences in males and females in terms of overall brain size. And so then we wanted to look and see, well, does this increase in progenitors actually then lead to an increase in neurons? And indeed, it does, and this is also quantified here. But in particular, it leads to an increase in the excitatory neurons produced within the dorsal cortex. And this is our model where essentially we've shown that if we treat these organoids with androgens, we get an increase in the proliferation of these progenitor cells that give rise to the intermediate or basal progenitors, and an increase then in the number of neurons. And this increase, particularly in the excitatory neuron production, is also very interesting from the standpoint of autism spectrum disorder, where it's also thought that there may be an imbalance in the excitatory to inhibitory balance. And so potentially this male bias could be coming from the role of androgens on these on this excitatory neuron production, which might essentially create a slight increase in risk, which perhaps in combination with other risk factors like genetic risk factors or potentially environmental, basically push it over the threshold, and then you start getting symptoms. So with that, I want to say thank you to everybody that was involved in this work. I think I highlighted Eva quite a bit. She led all of this sex bias, sex differences work that I showed you. And I also showed briefly some of the work from Laura Pellegrini, particularly the work on COVID and choroid plexus organoids. And also, I want to thank especially Magda, who is kind of the lab manager who runs everything very nicely in the lab for us, as well as, of course, my collaborators and all of my funding. Thank you.
Thank you so much, Madeline. We have time for one question. And if I can ask the next speaker to bring up his slides in the meantime. First question is by, oops, now all the questions are coming in. Fabio Cavaliere, congratulations for your impressive results. I concur. Can you comment how a blood vessel system may impact your results, especially concerning sex? Okay, so, well, a blood vessel system is obviously missing from organoids, and so that is limiting the degree of maturation, size,
Of course, in vivo, we would normally be getting vascularization. But interestingly, vascularization actually doesn't happen until quite later than a lot of people realize. So for about the whole entire first trimester, the human brain is actually not vascularized. So it's just getting nutrients from the vasculature over the surface and obviously the ventricular system. And a lot of the events that we're looking at are so early, actually, that it would even in vivo be before the vascularization would be playing a role. But I think it'd be really interesting to look at that in the future.
Okay, we actually have time for more questions until the next speaker is setting up. In relation to the COVID data, which cells are expressing Claudin-5 in the organoids? Is it known as a brain endothelial marker, but appears in your choroid plexus?
I mean, Claudin-5 is a tight junction marker that's also shown in the choroid plexus. So it is more widely known in the blood-brain barrier, but I think that's just because the blood-brain barrier gets more attention than the choroid plexus does. But the choroid plexus has just as tight a barrier as the blood-brain barrier does.
Okay, maybe one last question by Jose Ordovas Montanes. Do you think that similar mechanisms of androgen-induced proliferation of progenitor could be at play in some of the peripheral nervous system changes in autism spectrum disorder, or are these unrelated processes?
I actually hadn't thought of that one. That would be very interesting to look at. We do get in our organoids, we do get some neural crest, and of course that would give rise to peripheral neurons. But we haven't looked at those at all in our treatments, so that would be worth looking at.
Okay, just Croft is asking, are the connections from cortex to muscle cholinergic?
So we didn't specifically test. So basically the connections from the spinal cord to the muscle are provided by the mouse and are already, the mouse tissue, and are already in situ. So we're not really modeling that part of the circuit. We're modeling the upstream part. So basically the cortical motor neurons that are innervating the spinal cord. Now those are glutamatergic, and indeed we can show that, for example, we put glutamate on, we'll see an increase in contractions. If we put tetrodotoxin on, we block it.
Another question by just Croft, are fetal gonads or local synthesis producing androgens, estrogens in male versus female embryos at this stage of development, or is this dominated by maternal hormones at this stage?
Yeah, so I mean, I'm basing this on what's known from in vivo work. So obviously we haven't looked at those kinds of things in our in vitro work, but in vivo it's pretty well understood that actually the embryo, male or female, is sort of a protected environment from the maternal hormones, and this is through the action of alpha-fetoprotein, which actually essentially wipes it clean. So then what happens is female embryos essentially form in the absence of hormones, whereas male embryos form in the presence of testosterone. So this is why it's often thought that sort of female is the default. And so it's really the testosterone being produced by the embryo itself that's helping, that's leading to proper gonad formation and genital formation, but also impacting the brain, we think.
Okay, thank you so much, Madeline. We are now moving on to our next and last speaker.